When collecting evidence from a crime scene, the type of DNA and collection method will influence the DNA recovery success rate depending on the object. Surface types are categorized as flat or rigid, and porous or non-porous. These primary surfaces consist of fabric, wood, plastic, and glass. The recovery of DNA is influenced on the type of collection surface. For instance, wood can recover up to 169 nanograms of DNA, while fabric recovers up to 14.8 nanograms and glass can recover only 5.2 nanograms. Therefore, it is important to consider these types of surfaces and how much DNA is recovered prior to the collection process. With the use of hotspots, investigators can collect samples from areas with known high levels of DNA in order to maximize DNA recovery. For instance, in fabric gloves, the inside surfaces of the finger pads are the best spots to recover DNA, versus in latex gloves where the entire inside surface is a hotspot. When collecting DNA, investigators frequently use the two-swab method, which consists of a wet and dry swab. However, recent studies found that the use of a single wet swab is best for recovering high levels of DNA. And swabs are best for collecting from non-porous surfaces as well as collecting fluids. Adhesive tape is the second most common collection method, which is more suitable for collecting epithelial cells. However, there is a threshold to the tape's effectiveness, and if the threshold is exceeded, then the DNA recovery percentage decreases. The third most common method is cutting material, which is a destructive method and should be used sparingly as it provides similar recovery results to less destructive methods. And if the location of the DNA on the object is unknown, then cutting material can lead to contamination or DNA loss. After selecting the appropriate method based on the collection surface, then you can proceed with collecting the evidence. If the sample is wet, then the evidence must be dried before it can be packaged. Otherwise, wet samples can lead to contamination, bacterial growth, or DNA degradation.